your bone. Let me talk about radon gas. If you Google radon gas, that's what you're going to get. Remember, radon's the gas that comes from radioactive decay. That's a picture of radon gas. That's a picture probably of me when I lived in Elliott Lake, because they told me my house was full of radon gas and I was going to die if I didn't stay out of the basement for more than 2 hours a week. It was going to be a big dose of radiation to me. So unfortunately, my bedroom was in the basement, so I got a bit of an issue with that. So probably that's why I'm standing up here today. A little bit of misinformation always inspires people to do things, and that definitely inspired me to figure out whether these guys knew what they're talking about. But there's still a debate whether or not radon gas causes cancer at high doses of it. I mean, high doses, it would seem, yes, there is an increase in cancer incidence, but what about at low doses? Even the high dose stuff is controversial, but at low doses, what about low doses? Well, let's look at, there's one study that looked at low doses, this one here, by Bernard Cohen. Still hasn't been challenged. I like showing it because it simply shows that when you look at lung cancer deaths per 10,000 people and correlate it to radon in non-smokers or in smokers, if you didn't have any radon gas at all, you'd expect there to be a flat line, right? If radon gas had no impact on lung cancer, you'd expect a straight line. If it was related, which it looks like it is here, as the radon in the low dose range concentrations go up, the probability of dying of lung cancer goes down. So it's an inverse relationship. So that's sort of saying at these low doses, having a negative risk, because it would be normally 7 under normal circumstances, but now we're dropping it half almost as we get up to these low levels of radon in your house. And then at some point, the authors argue that it's going to go up again, because we're getting to overload the system again, and it goes up, and then at some high dose you get it back up to where you were before, and then it's going to increase above that.
Sorry, just so I understand that. That's showing a decreased incidence of lung cancer deaths with increasing radon concentration. At
low doses, yeah. So this is picocuries per liter. As you go up to about 5 picocuries per liter, the risk of dying of lung cancer is a lot less. This is in 1,600 different counties.
Does that relate at all to working
This is just in houses, in your basements houses. People exposed to natural radon stuff.
So the increase is they're not showing any actual increase.
No, there's a break in that data, which is why they're predicting, because they know in the higher doses way out, when you go way out, okay, when you're uranium miners, that there could be increased risk. Somewhere it's going to get back up. This is just the real data. This is about as far as they get in some of their collecting of their data in the houses. And as far as I know, it still has not been related to anything else other than radon. There's about 2,000 different confounding factors you can look at here, but radon seems to be the direct correlate of risk of dying of lung cancer. And the biggest thing is, I think, when you're a smoker, your risk goes up, but this impact is even greater in smokers. Less lung cancer risk in smokers if radon gas is higher. And of course, people want to take advantage of these things, so you buy an old uranium mine in Montana, you charge people to go in it. I think they charge about $100 to go in once a day for 2 hours, sit there and breathe radon gas for an hour a day. And it works. Well, there's all sorts of mines, like there's this place in Europe, they 10 treatments every other day. They are at 4,500 picocuries per liter. Remember the other graph showed only up to 5? So they're pushing it here. Okay. But these people believe, and there is evidence to suggest that these people are not being harmed by it. So people are using this as a therapeutic thing. They've been doing this for years. All right, I'm going to go back into find out the mechanisms of what we're doing here, because I got to wrap this up soon. At McMaster, we're approaching this in sort of 3 different ways. We're trying to understand what the risk is when you are exposed to the low doses, not the high doses and extrapolate down. We're also trying to figure out what the relative role of your genetics and environment are, and we're also looking at ways of detecting whether or not people have been overexposed to big doses using tools that we're developing here at Mac. So in other words, when Fukushima happened, we got a phone call from Health Canada saying, how many samples could you process at McMaster University if we were to get hundreds or thousands of samples from Japan, from people in Japan that were possibly overexposed? And our number here is we could probably process about 600 blood samples a month if we ramped it right up to look for changes in chromosomes that would give us a correlation to if you were exposed to external radiation. So we're developing these kind of things. We never got any samples from Japan because no one actually got a big enough dose for me to detect, even though we were told a lot of people were getting it. No one ever got a dose higher than, as far as I believe, somewhere around 150 millisieverts over the 5 days or 6 days. And they're allowed during emergency situations to get somewhere around 200 to 250 millisieverts to do emergency operations. So no one even got that. So that was a good thing. Although people were told that this was going to be a big thing, it never ended up. It was handled in a way that people weren't overexposed. Because you can easily detect the stuff, you can measure it, you can monitor how much you're going to get, and then you can get out when you've had enough. All right, so just basically the types of radiation we're exposed to. The radiation itself, you know, when I'm talking about alpha particles and beta particles and gamma rays, these are energetic photons or particles that have virtually the same sort of impact when they hit things. They create free radicals. They ionize our molecules to different extents based on the physical characteristics of them. But the bottom line is, depending on where they come from, that just basically tells you what type of particle they are. Alpha particles are simply helium atoms, beta particles are electrons, and they come flying out of unstable atoms and interact with us. This is just showing you an example of an alpha particle. If you look at an alpha particle in what we call a cloud chamber, you can actually see the track of radiation moving through, and it creates these ionization as it goes. So it's a very dense track along its main path, but you see all these little spurs coming off, these other little tracks, and that's just scattering radiation. So that's what your cell's going to see as an alpha particle goes through it. And so this is just a cartoon showing you that all these things, these little dots, represent an atom being ionized as this particle goes through our cell and our DNA. And if it interacts with our DNA, this is a model of a double-stranded DNA helix. You can get an ionization on a base. You can ionize the backbone of the sugars that cause damage to DNA. Or if you have a highly charged alpha particle, it does base damage and both strands get broken, called the double strand break, which is why these higher energetic particles like alpha particles will cause more damage to DNA per unit dose delivered to the cell. That's why they become more biologically damaging when they have higher energy deposition per unit track length. But at some point, if we look at what happens, again, this is a DNA strand. This is what we call relative biological effectiveness. As the energy that's transferred as the particle moves along that track goes up, so as LET goes up, the RBE goes up, until it peaks at 100 kiloelectron volts per micrometer, then it drops off again. And the reason for that being is that the double strand break in DNA is the critical thing that causes issues with us when we get too many of them. And so at this energy level, that's roughly where you get an ionization occurring at the same distance that your DNA is, which is roughly 20 angstroms. So it has the maximum biological effectiveness. After that point, the RBE starts going down based on the energy absorbed, because you can't get more than one double strand break. You're just wasting energy. See what I'm saying? So that's why when we talk about alpha particles and other things, they will cause double strand breaks, but there'll be a lot of ionizations there that won't have an impact because they're basically just gone through the cell. It's a waste of energy. It's not causing a double strand break. So when you're thinking about the particles you might encounter while flying, the alpha particles, protons, they have this higher LET that basically is going to start dropping off as you get, the effect will start dropping off as you get higher and higher energy of them. So it's just a way of saying it's not all bad. As the energy gets higher, it's not all bad from a DNA perspective. All right, I'm going to bypass this because we're running out of time. This is my little model of what happens to cells when they get hit by radiation. So if you start with a normal cell, and pretend this is the nucleus of a cell where all the DNA is, where all those strand breaks are going to happen, and you run a photon or alpha particle through it, it's going to cause damage to the brain of the cell, the nucleus. And three things can happen. One is the cell itself will be able to repair itself to normal, which means the consequence of the photon going through the cell is not there. It's repair back to normal, no consequence. On the other hand, this could kill the cell. The cell dies. Well, there's a consequence to the cell, but there's no consequence to the organism, because dead cells can never become cancer cells. So dying, a single cell dying is a good thing, and single cells repairing themselves back to normal is a good thing after being interacted with radiation. The problem we get into is this, is when you get an abnormal cell, and that is, I've had this sort of personality changing of the cell where it actually doesn't do what it's supposed to do anymore, it misbehaves, and that cell can then become a rogue cancer cell and cause problems in your body. But the real model is this. That's a little simple model. This is what we kind of use for the three pathway model. Damage the DNA of the cell, which is the brain of the cell, repair yourself to normal, or kill yourself, commit suicide. These two pathways are genetically controlled, modified environmentally, but work very hard to stop the cell from going down this pathway. And it's when you get genetic defects in either one of these pathways or environmental stresses or influences that change the way those pathways work is when you can drive it down this pathway. The point I want to make here in this slide is that radiation only starts at this point. There's many steps that go from here down to here before you get here that the LNT model does not take into account, and that we're learning the impact of these pathways has on all the risks of going down these different things. This is just a blood sample I take from a human. There's about 40 dots here. I don't know if you can see them. There's red dots. There's 4 yellow dots. These 4 cells have committed suicide after I irradiated this population of cells so I can count them. They're very elegant tools to tell which cells are committing suicide. I call it committing suicide because it's called programmed cell death or apoptosis. It means the cell has made a genetic decision to eliminate itself from the population. It's not just falling apart. And the DNA repair processes, we know there's some 300 or 400 genes that control that pathway. A single defect in one of those genes in that pathway can cause severe increase in cancer risk going down this pathway. So a couple of experiments I'll show you about and then we'll be finished. Well, cells have to go, like I said, once they get initially damaged by something like radiation. If there's too much of it there, you get this damage will actually persist enough that the cell says, okay, I've got some genetic damage here that's going to cause a change in the way I do things. You have to be able to measure that using different things. And when a cell changes, we call it the cell is being transformed from a normal cell to a cancer cell. So the assay is called the transformation assay. And this transformation assay is an example here of some cells in a flask. Some of these cells, when I put 10,000 cells into a flask like this, some of these cells will turn into cancer cells, depending on what I do with them. They transform. So instead of doing what they're supposed to do in this flask, they will actually invade the flask and take over like a tumor would in your body. Start growing out of control, growing fast, and invade. And this is kind of an example of one here. So we put 10,000 cells in the flask, we wait 6 weeks, we feed them, we look after them in incubators, and we ask how many cells in the flask turn into these transformants, or precancerous cells. And they kind of look like this. You can see this one here up close. It's a lot of cells piling up on top of each other and invading out into the flask, so taking over the flask. And that's what tumors are. They pile up, they have out-of-control growth, and the key thing about them is they invade, and they interfere with other organs and other tissues. So we call that type 3 foci. If we take the cells from this and inject it into a mouse, we know they cause cancer because they will kill a mouse. It will cause tumors in them and kill a mouse. So we just count the number of these foci we get in a flask, depending upon the treatment, and ask, what was the risk of transforming cells in the flask? It's a very simple model system we use. Well, if we do nothing, and I'm using milligray here, just think millisievert, 1 millisievert, 10 millisievert, 100 millisieverts. If I do nothing, about 18 of these cells out of 10,000 will turn into spontaneous transformants. That's what we call the spontaneous rate of cancer. And if I expose the flask to 1 milligray, 10 milligray, or 100 milligray, the number of cells that transform actually drops. Does not go up in this low dose range. They don't get killed, they actually eliminate themselves. So what it's saying is, at these dose ranges where these are single acute doses, either 1, 10, or 100, it actually reduces it roughly to the same extent. So it's like an on-off switch. It's not proportional to the amount that you give it. You give this little dose, you actually reduce the transformation frequency. That's been known for many years now, and a lot of other studies have reproduced that in different model systems showing that these low doses of radiation somehow have an anti-transformation or anti-carcinogenic process involved with them. There's a study done in California, same idea, different scale. I should change that, but if you do nothing, the number of transformers is about 3 per 10 to the 5, and these low doses actually drop it, and as you get higher doses, it starts going up again. It's a classic sort of curve I was telling you about, the U-shaped curve that decreases risk at the lower end and increases at the higher end. We've shown this in mice for cancer risk, where we've looked at mice that we've given a big dose of radiation to, so 1 gray or 1 sievert all at once. These yellow dots represent the mice that die of leukemia with time, so they're almost all dead by 600 days. This other group of mice got exposed to 100 milligray or 100 millisieverts 24 hours before getting the leukemia-inducing dose, which is this one, and they lived about half the time of normal mice. So they extended their life by half, which again is a positive effect in the sense that these mice lived significantly longer by getting a little more radiation. It just happened to be a low dose before the big cancer-inducing dose. So again, some more evidence suggesting that maybe these low doses aren't causing a negative effect and perhaps are modulating the biological processes that are involved in risk. I thought I'd put this enough for breast cancer because that's one of the risks associated with being a flight attendant. Not from radiation, but from whatever else causes it, mostly not having children, I think. It's a small risk, like I said before, but if you actually look at radiation and breast cancer risk in people that were given fluoroscopy treatments in history, you can see that this is centigrade, so add a 0 and you'll get milligray. 50, 100, 200, 500. These were given in multiple series, so not all at once. So doses were given every couple of weeks to follow tuberculosis management in these patients and women in Ontario and Canada. And it shows, again, that this is what it is if they didn't get any radiation. And as the radiation dose goes up, we have that drop at the low end, and then again, it gets higher again. So again, a non-linear response with some kind of threshold. And I think that's about all I want to say with respect to that, except a paper just came out, well, last year now, almost a year and a half ago, where they're asking about babies being exposed to radiation. So there's, again, this issue about pregnant women flying or pregnant aircrew flying, getting exposed to cosmic radiation, women that may be pregnant working in the nuclear industry. Is there an increased risk to them? Well, using the precautionary principle and the ALARA principle, we say yes, there could be an increased risk because you have a developing fetus. There's many rapidly dividing cells there. There's a chance that if we expose those rapidly dividing cells to a carcinogen, it could transpire into a carcinoma someday down the road. There's some evidence to support it. It's not very strong. In fact, there's some evidence that says that that's not the case at all. But to be safe and to be cautionary, we just say, okay, let's reduce the risk as below is reasonably achievable and not expose so-called pregnant women that are, especially fetuses in the first trimester of development, to any excessive radiation that they don't need.